Hello, everyone. I welcome the students, researchers, scholars, and scientists all over the world. This is Advanced Multiple YouTube channel. Advanced Multiple Incorporation actually a knowledge processing and information management organization that is located in Mississauga, Ontario, Canada. My name is Dr. Taha Nazir. I'm working as scientific executive with the Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Okay, well, today I'm going to share my knowledge, my information, my experience for my research under the title of Introduction and Methodologies Used for Immobilization of Enzymes. For this purpose, I collected the scientific research information from different open source like Medline, PubMed, Scopus, Google Scholar, National Library of Medicine, US, MBES, directly of Open Access Journal, ProQuest, CNKI, that is China-based database company, provides scientific research information all over the world. CNK stands for China National Knowledge Infrastructure. Some of information collected from the ResearchGate and Crossref. Okay, well, before moving forward, I would like to talk a little bit about the introduction of immobilized enzyme. Immobilized enzyme is an enzyme attached to an inert, insoluble material, most of the time calcium alginate. Immobilized enzyme produced by reacting a mixture of sodium alginate solution and enzyme solution with calcium chloride. Immobilized enzyme can provide a resistance to change in condition, for example, pH, temperature. Immobilized enzyme retain the catalytic activity and can be used repeatedly and consistently or continuously. It is a process of confining the enzyme molecule to a solid spot over which a substrate is passed to convert into the desired pharmaceutical, chemical, or health products. Okay, well, there are the different kind of the method used for immobilization of the enzyme. Immobilization meaning converting the mobilized enzyme or the unstable enzyme into the immobilized enzyme or the stable enzyme that is able to fight against undesired, challenging situation or condition. There are the two main methods used for immobilization of enzymes: physical and chemical. There are the three further subtypes of the physical method: adsorption, entrapment, and encapsulation. In chemical method, there are the two categories: covalent binding and cross-linking. Okay, well, adsorption physical method involve the physical binding of the enzyme on the surface of the carrier matrix. Carrier may be organic or maybe inorganic, for example, silica, bentonite. The process of adsorption involves the weak interaction like van der Waal or hydrogen bonds, for example, catalase and invertase. Entrapment physical method, the enzyme are occluded in natural or synthetic polymers. It is permeable membrane that allow the substrate and the product pass but retain the enzyme inside the network. It can be a gel or the bead entrapment. Entrapment in calcium alginate is most widely used in actual practice. It has one disadvantage, that is enzyme may leak from the pores. It involves entrapping enzyme within intestinal space for the cross-sectional cross-linked polymers. Synthetic polymer, for example, polyvinyl alcohol, and natural polymer, for example, starch used in immobilized, the enzyme being used for this technique. Encapsulation physical method, it involved enclosing the enzyme within semi-permeable polymer membrane, semi-permeable nylon membrane in the shape of the sphere. Okay, well, covalent binding chemical method, enzyme form covalent link with the active group of the matrix. The functional group of the enzyme which is involved in the linkage should not affect the active property of said enzyme. The functional group that may take part in this binding are amino group, carboxyl group, and hydroxyl group. But covalent binding may alter conformational structure and active center of the enzyme. Okay, well, cross-linking chemical method of immobilization. This is the last method that is used for the immobilization of the enzyme. So cross-linking involve intermolecular cross-linking of enzyme molecule in the absence of the solid spot. The method produces three entities: dimensional cross-link enzyme, then aggregate insoluble in water by means of a multifunctional entity reagent that link covalently to the enzyme molecule. Okay, well, this is a typical model of the bioreactors and modes of immobilization. It comprises of the two main components. The upper component that comprises of the four bioreactors used for the immobilized enzyme, and the lower segment that comprises of the mode of enzyme immobilization for the respective bioreactors. There are the four main type of the bioreactors: packed bed reactor, fluid bed reactor, then retentostate membrane reactors, and there are the five different kind of the modes of the enzyme immobilization involved. The first one is the cross-link enzyme aggregate, then immobilization on solid support, entrapment of the matrix on gel. Fourth one is the permeabilized cell and semi-permeable membrane. The last one, fifth one, is the lactase immobilized membrane. Okay, well, in conclusion, the performance of immobilized enzyme greatly depend upon the immobilization method and carrier type. The carrier enhance the biocompatibility, thermal stability and insolubility under challenging industrial and environmental conditions. The regeneration and reusability of the enzyme also determine the cost, quality, and efficiency of the final product. So, based upon recent scientific advancement, a novel and resourceful and efficient product can be produced in dairy, pharmaceutical, and chemical industry. Uh, okay, well, before closing the presentation today, I would like to salute and greet to my colleague and my co-worker, my team: Sherry Bachkat, Wolfgang, Daniel, Dustin Hawker, Marwa Al-Maghribi, then Robert Heyman, Amin, Arslan, Ishtiaq, Harry Mill, then Faisal, Noreen, Kunza. They helped me a lot to make this project successful. I also acknowledge the kind help and support of Advanced Multiple Incorporation and the Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Thank you very much for staying with me and watching the video, spending a very nice, beautiful time. I hope I will see you soon in my next session, in next video, and take care. Bye for now.